Hey everybody, Professor Carafit here. If you missed the natural selection lab due to an excused reason, you can make it up by watching the pre-lab video that was already posted for everyone on the lab website. If you forget what that is, it's uca.edu/go/bio2, or you can get to it through the lab website. And then you need to watch that pre-lab video, and then watch this video, and you'll be given a data set to go through. And also, please review this lab in the manual. I'm knocking stuff down here. That's lab 3 that starts on page 21 of the manual. You can see what everyone did in class. So we began the pre-lab talking about natural selection. We did an example using playing cards where everyone pretended to breed with one another and mix up our alleles, and we looked at the effects of genetic drift on our genotype values, and we ran them through the Hardy-Weinberg Equilibrium. And then we moved on to the natural selection experiment, which is where I'll pick this up. So organisms tend to be able to hide at least a lot of prey organisms and some predator organisms due to their cryptic coloration, their ability to blend into their environment. Here we see a fence lizard. Here we see some inchworms. They look like sticks, these caterpillars hiding on twigs. Here is a flounder, which obviously doesn't hide on a checkerboard, but hides on the sea bottom where there's a series of light and shadow, thanks to the coral reefs and things that cause light and shadow on the bottom. Here we see the fall foliage and a snake that's found all over the place here in Arkansas. That's a copperhead. You can see how well it blends in. Eggs can also be camouflaged. Now humans kind of lack this cryptic coloration, but we can use our our minds and our imaginations and our artistic abilities to blend in. Here's another example of a fish camouflaged on the bottom of that coral reef. So we're going to look at how that variation may have come about in the experiment this week. And I'll show you one more example here before we do that. The peppered moth found in England. This is a very common textbook example of microevolution. So the peppered moth came in two forms, white and black. The white variety was more common before the Industrial Revolution. Then the Industrial Revolution came along and trees got covered in black soot and things from the coal-burning fire, the coal-burning plants. And then the white moth stood out, but the black moth became more common. Then later on, environmental policies changed, and the factories cleaned up their act, and the tree bark started going more white with more lichens on it again. Now the white moth is much more camouflaged than the black moth again. So we've been able to see this change, this evolution over time. So in your lab manual there is a worksheet. So the way this lab was conducted, there is a tub of little tiny red beans, and that's the habitat. Meanwhile, there are 3 kinds of big beans that students are given: white beans, red beans, and mottled beans, which are red and white. So we start off by assuming that the red allele, that's R, or color red, R, is 50% of the population, 50% of the gene pool, and the white allele is 50% of the gene pool. Then we run that through Hardy-Weinberg Equilibrium, which is kind of broken out here like a table. So red times red, 0.5 times 0.5 is 25%. That means 25% of the population so we're simulating rabbits here. So the beans stand for rabbits. So 25% of the population would be red, 50% of the population would be red-white, and 25% of the population would be white. Okay? And so what we do then is we assume the carrying capacity of the habitat is 60 rabbits. So we multiply these proportions by 60 rabbits, and we get that we should put 15 white rabbits, 30 mottled, and 15 white into the habitat. Then students are given 20 seconds to find as many rabbits as they can. So in this example, the students remove 3 red, 7 mottled, and 6 white, and they figure out how many survive. Okay, so total survived were 44. Then what the students do is they figure out, okay, if there's 44 surviving rabbits, that means there's 88 total surviving alleles. And so then what they do is they figure out, okay, well, how many of each allele survived this predation event. So you take 2 times the number of red survivors. So in this case, 12 times 2 is 24 because there's 2 red alleles in every red rabbit. So there's 24 red alleles that survived from red rabbits. And within mottled rabbits, that's red-white rabbits, that's their genotype, there's 23 red alleles. So you take 24 plus 23, that tells you there's 47 surviving red alleles. You do the same thing for mottled, or for white I should say, and there's 41 surviving white alleles in this population. Then you divide them by the total number of surviving alleles to get their proportion. In this case, 0.53 for red and 0.47 for white. Then you plug those numbers into the next generation. Generation 2, you do the math, Hardy-Weinberg, and you figure out how many rabbits of red, how many of mottled, and how many of white to put into the habitat. And then the next lab partner was a predator for 20 seconds and did the same thing. So this went on for about 6 generations, and that's the data you'll be given, is the results of that experiment. Okay. So then the experiment was restarted again at 0.5.5. So just right here, 0.5.5, P and Q is 0.5.5. It was restarted and done again. Only this time students were given 1 minute and they had to close their eyes. So I sent you already hypotheses for today's experiment. So you'll use those when you're trying to write up your assignment. So let me talk about that assignment. So what I'm going to do here is try to create a blank slide that I can draw on. We'll see if this will work. Sometimes it works, sometimes it doesn't. So, right. So your assignment is going to be, first of all, to write a results section of a lab report and a conclusion of a lab report. So the results section will include 2 figures. Figure 1, let me see if I can trick this thing into working. Come on, buddy. This is a pain to do this live. All right, there it is. All right, don't worry about that. All right, figure 1 is going to be a scatter plot with generations down here and P over here. P is the proportion of the red allele. And of course, it's going to start off at 0.5 because that's how we started both blind predator predation and visual predation. And you're going to graph what happened to the red allele with both blind predators and visual predators. So there'll be 2 lines there. And you're going to have a very simple caption. You'll just have the figure number down here and a sentence, title sentence. You don't have to quantify anything. There are no error bars on this graph or anything like that, okay? That's the first graph. The second graph is going to be a column graph with error bars, with a caption, with a t-test and all of that stuff. And it's going to be the, so let's just say visual and blind. I'm writing with a mouse, so I'm not going to go full out here. Visual and blind, and this is going to be number of rabbits eaten per generation. The average number of rabbits eaten per generation, total, no matter what color, by the visual predator and by the blind predator. And that will be figure 2. Then you're going to write a discussion section where you see whether or not the hypotheses were supported. And then you'll also, within that section, do everything you need to do for a discussion as described in the lab manual in How to Write a Lab Report, which is Appendix B that starts on page 113. You're going to do all those things, and you're also going to have a literature cited section at the end where you can make sure you cite your textbook and cite your lab manual. That's all I require for this assignment. If you've not written a lab report before, this might be new for you. I have a video on how to write a lab report on Blackboard in the lab report folder, which is found within the lab folder. So that is your assignment, and it's due 1 week from the time you have the natural selection lab, or if you missed, 1 week from the day you would have had the natural selection lab. Okay? If you have questions, come to my office hours. We can sort it out, and we'll talk about it then. I hope you feel better soon. Bye.